Okay, Assalamu alaikum everyone. My name is Areeba, and today we are going to be discussing blood gas transport, right? So, what are the two important gases in blood? There's oxygen and there's also carbon dioxide. So, we'll start with the big one first. We're going to start with oxygen, okay? So, oxygen in our body, it's present in two forms. There is a physically dissolved form, which is basically just the free oxygen molecules. And we also have chemically bound oxygen. So bound to what? They're bound to the hemoglobin in the blood. And just so you can visualize it, it's this way. So you have these little red dots, right? You see these. These are all the free molecules. And this is a red blood cell with hemoglobin. And these tiny little dots are just the oxygen molecules bound to it. Okay, so starting off with the physically dissolved oxygen, let's take just a little environment. We have gas, which has oxygen, obviously, and we have a fluid. It's any solvent, okay? So the pressure that the gas is going to exert is going to make the oxygen go from the gas into this fluid, right? That's the physically dissolved. I mean, as the name says, it's just oxygen from the gas which gets dissolved into this fluid. Okay, oxygen that is dissolved in the blood obeys Henry's law. So, what is Henry's law? It's this simple. It's a law that states that the amount or the mass of a gas in any given volume of fluid or solvent is going to be proportional to the partial pressure of the gas. So if we know the partial pressure of the gas, we know how much pressure this gas is exerting, we can kind of make out how much of that gas is going to be present in the fluid as well. So the amount of oxygen dissolved is proportional to the partial pressure and the solubility. Because other than the pressure, if your fluid, if your solvent is not readily absorbing the oxygen, that is also going to affect how much oxygen is going to be there, right? So there's two things, partial pressure and oxygen solubility. So this is just the equation showing the Henry's law. Its concentration of O2 is equal to solubility coefficient of O2 multiplied by the partial pressure of O2. Okay. If we look at the physically dissolved oxygen on the graph, we see that it's a very, like this line is kind of low, right? So even when we have a really huge pressure difference, if you look at it corresponding to the oxygen concentration, it's not increasing that much. So even if we go from, let's say, 150 to like 225, it's a very, very slight increase, right? But in our body, physically dissolved physically dissolved oxygen concentration is very important because physically dissolved oxygen is the way that oxygen will be transported throughout the body. Because this is the free oxygen molecules. This is the only way that the oxygen is going to be able to diffuse from membranes. So when you breathe in, the oxygen, for the oxygen to go from the alveoli to your capillary, it has to be in the physically dissolved form. Okay? So if we look at the amount, it's not that significant. And the amount that our body uses is a really huge amount. However, this is the only way that oxygen can be transported. Okay. So we looked at physically dissolved oxygen. The second form of oxygen was the chemically bound, sorry. So for chemically bound, this is what the graph is going to look like. And we see that it's actually really, really huge. So a very, very large portion of our body's oxygen is present in the chemically bound form. So O2, it reversibly combines with the hemoglobin and it produces oxyhemoglobin. And oxyhemoglobin, it's just hemoglobin with the oxygen. And if you compare the small physically dissolved part, this little blue area in the bottom, to this little, the orange area, you can see how much is chemically bound, right? So that's basically what this thing is representing, this whole thing. It's showing you the difference. So basically the difference between this blue area below and this. This is going to tell you how much is chemically bound inside the body. The oxygen concentration in the blood is related to the partial pressure of the blood. We saw that in the Henry's law as well. And another thing we can see in this graph is the O2 capacity. So what is O2 capacity? It is basically when all the binding sites on the hemoglobin are occupied by oxygen, we say that we have reached O2 capacity. Okay, so chemically bound, we're saying that it binds to the hemoglobin. And in our body, hemoglobin is only limited to the amount of blood we have in our body, right? So at some point, as it increases and increases and increases, it's going to reach this point, and now you have reached a point where all your hemoglobin is used up. So after this, at this point, you can't bind anymore to the hemoglobin. So you've reached O2 capacity, which is the maximum chemically bound oxygen. So if we reach this capacity, we reach O2 capacity, and you add more oxygen to the body, where is it going to go? It's going to start adding up in this area, in the physically dissolved oxygen. Okay. And this difference, you see this difference from here to here, this is going to stay the same throughout. So basically this blue line is going to run parallel with this one, and whatever extra oxygen is going to come, it's going to be added into the physically dissolved area. Okay. So we said the binding of hemoglobin. So this is basically just to show you that hemoglobin has binding sites for oxygen. And if all of these are just taken up, then we say that, okay, it's saturated, it's completely occupied. Okay, so we have physically dissolved and we have chemically bound. And now we're going to see how they act with each other in the body, okay? So we're going to take a little system, a little experiment almost. So we're going to have two chambers of gas, and they're going to be separated by a membrane that is permeable to oxygen molecules. Okay. Now, in both the chambers, we're going to put equal amounts of oxygen molecules. So we have 8 in A and 8 in B, and the pressure is going to be the same, obviously, because we have equal amount of molecules. So one important thing you need to remember is that pressure that is exerted, the PO2 that we're talking about, it is only produced by free oxygen molecules, okay? So when we're talking about the PO2, it's basically the physically dissolved oxygen that exerts this pressure, okay? Now, in chamber B, we're gonna introduce something that can bind to oxygen, and in our case, we're gonna take hemoglobin. So hemoglobin, when we put it in chamber B, these red molecules, these oxygen molecules, they're gonna go and they're gonna bind to this hemoglobin, okay? Now, like we said, pressure is only exerted by the free molecules. So when these oxygen molecules, they bind to the hemoglobin in chamber B, you're gonna be left with only two molecules. What is it gonna do? It's gonna decrease the pressure in chamber B, right? Now, if there's a pressure difference, this is what's going to cause the movement of oxygen from one place to the other. So you know how we have concentration gradient? So it would be if there were like more oxygen molecules in one place, it goes to where there is less concentration. Here it's not concentration. Here the movement is primarily dependent on the difference in the partial pressures. So because it's just this one and this one, now the oxygen molecules are going to start moving from A to B. What happens? If the molecules were just going to move from A to B, we should have just seen it become equal and hemoglobin with the three oxygen molecules. However, we see that hemoglobin has even more oxygen molecules bound to it now, and there's two on the left and there's two on the right. So what happened is when the oxygen molecules were moving from A to B, the hemoglobin here, it still had capacity to bind to the oxygen, okay? So whatever oxygen would come and try to increase the pressure, the hemoglobin would bind to it. Once the hemoglobin was fully saturated, now, after they were saturated, the only free molecules that were left are the ones that balance out the pressures, okay? So you have to remember it's not about the concentration, because if we look at B, there are so many oxygen molecules. However, the PO2 and the PO2 in B and PO2 in A are equal, because this one and these two are equal. Okay, so that was the O2 concentration. Now let's look at O2 saturation. So O2 saturation curve kind of looks similar to the concentration curve, but there are differences, however. Okay, so in the saturation curve, the y-axis, the O2 saturation axis, this 1.0 basically signifies 100%. So when we say 1.0, it says 100% saturation, okay? If we look at 50% saturation, when the blood is 50% saturated, and we look at what pressure that is, It's 27 millimeters of mercury, okay? This 27 millimeters of mercury is the P50, and P50 is an important measure for us because it measures the affinity of the hemoglobin for oxygen. How does it do that? Basically, it's saying that when the pressure is 27, that is when hemoglobin is able to bind to half of the oxygen that is available. So if the P50 was more, if the P50 was way higher, let's say it was somewhere like here, that would mean that you're requiring more pressure for the hemoglobin to bind, right? So that kind of tells us about the affinity of hemoglobin for oxygen. Another value that we look at is this 100 millimeters, and we have 40 millimeters. Where do we have 100 millimeters of pressure? We have it in the alveolar region and the capillary blood, right? So what's significant about this? It's that at this pressure, we have 98% saturation. Means 98% of hemoglobin is binding to the oxygen, which is good because in the alveolar regions, in the capillary regions, we want more oxygen, right? So there's full saturation of hemoglobin in these areas. And when we talk about 40 millimeters, what does this signify? That is the mixed venous blood. So mixed venous blood is coming from the right ventricle. Sorry. Okay. So mixed venous blood is coming from the right ventricle to the lungs, right? And if we're saying that the blood is coming after being used up in the body and it's coming to our heart, we would expect it to have less oxygen, right? But then when we look at the saturation, it says that it's 75% saturated, which means there's still a lot of oxygen in there, right? So why is that? It's because of two reasons. Basically, our body, different parts of our body uses different amounts of oxygen, okay? So if we take our heart, for example, our heart is going to use a lot of oxygen. Oxygen consumption is going to be high. And then when we look at the saturation of the blood that comes from the heart alone, it's going to be really low. However, the blood, let's say from the kidneys, is not going to be using up a lot of oxygen. It's going to be highly saturated. So when these two bloods come to our right ventricle, they mix up, right? So these saturations are going to mix up. And that's why at the end, you have an average of 75% O2 saturation. And also, it's good for us. It's good for us if we have extra oxygen in the blood, right? Let's say, for example, we want to exercise, we want to do something all of a sudden that would require us to consume more oxygen than we normally do. So we don't want to compromise our organs, we don't want to compromise anything in the body. So that is why venous blood having 75% saturation is good because we need this extra amount of oxygen in the case of emergencies as well. Okay. Is everything okay? Does anyone have any questions so far?
No, no questions.
All right. So we said that there is a difference between the O2 saturation curve and the concentration curve. So what is the difference? Basically, when we see the O2 saturation curve, it is talking about the ability to bind. It is talking about how much of the hemoglobin is bound to the oxygen. Okay. When we look at the concentration curve, it's talking about how much oxygen is there in the body. Okay. So let's say, for example, hemoglobin is halved. If the concentration of hemoglobin is halved, what's going to happen? It's not going to affect the saturation curve because even if there's half hemoglobin, the saturation, the ability of hemoglobin to bind at the certain pressures are going to be the same. So at 27 millimeters of mercury, we're still going to bind 50% of hemoglobin. However, when we look at the concentration curve, concentration, we said it's coming here. Because if we have half hemoglobin, obviously we're gonna lower and we're gonna be somewhere here. So the concentration curve is gonna decrease. Okay. So if anyone has any questions, you can ask me now because we're gonna move on to more details about the graph. No questions? Okay. I'll take that as a no. So we saw the graphs. Now we're going to see at the different factors that will affect the graphs, right? Because we have a lot of things that are always going on in our body. So let's understand the graph even better. This is a saturation graph, and the red one is the normal one. And we said this is our P50 value, right? And P50 value tells us what? P50 value tells us about the affinity of hemoglobin. So it's basically the partial pressure of oxygen that is required to achieve 50% hemoglobin saturation. Now, when our curve shifts to the right, we say that it decreases in affinity. Why would you say that? Because if we look at this value corresponding here, we're going to see that our P50 increased, okay? Which means that we need higher pressure for the hemoglobin to be able to bind to the oxygen. So decreased affinity, right? The hemoglobin is not readily taking it up. And vice versa for the increase in affinity. If the affinity increases, even at a lower pressure that corresponds to almost here, our hemoglobin is going to be 50% saturated, okay? So we're going to look at the effects of pH and CO2 first. So this is the graph for pH, this is the graph for CO2. One thing to see is that in the pH graph, we're keeping the pressure of carbon dioxide equal. So that's solely the effect of pH. And then when we look at the effects of carbon dioxide, we're keeping the pH constant, okay? So what is pH? pH is the concentration of H+. As the concentration of H+ increases, the pH decreases. We know that it's gonna become more acidic. And when the H+ increases, the affinity of hemoglobin is gonna decrease. Why does this happen? It's because the hydrogen ions will go and bind to a part of heme, okay? So you see here, when the hydrogen ion will come and bind, this oxygen is left alone, and this site is left empty, okay? So the affinity decreases. The opposite is true for decrease in H+. So if it becomes more alkaline, if the pH increases, you have less H+. So this is not going to happen. So affinity is going to increase. And this is known as the Bohr effect. We'll look at Bohr effect more later, so don't worry about that. Now, carbon dioxide, it has two mechanisms. It has a direct mechanism and an indirect mechanism. The direct mechanism is when the carbon dioxide itself will go and will directly bind to the alpha and beta chains of the hemoglobin. So again, like we saw here, the space was left empty. This space is also left empty because the affinity is lowered, it's not taking up the oxygen. And this is where the carbon dioxide bound to the hemoglobin. Indirectly, what it does is it increases the H+ ions. So as CO2 increases, it will increase H+ ions as well. And like we just saw above, increase in H+ is going to do what? It's going to decrease the hemoglobin's affinity to bind to oxygen, okay? So since we talked about Bohr effect, let's look at that. So look at this equation. It's basically carbon dioxide and water is going to give us bicarbonate and H+, okay? You have to remember this. So H+, what it does is it goes and it binds to the hemoglobin, and it will induce conformational change. What is conformational change? It basically just changes the shape of the hemoglobin such that oxygen is not going to bind. So you see how it's like readily sitting over here, but here it's so thin, it's not allowing it to go inside. So when PCO2 is high, it's going to react with water. So this and this is going to give us H+ and bicarbonate. This is how it indirectly affects the curve as well. So the H+ will go to the red blood cell, change its shape, lesser affinity to O2, and the CO2 and water are the ones that react to give us H+ and HCO3, okay? This one is just the combined effect. Basically, both PCO2 and pH are affected, and this is how they shift. So increase in CO2 concentration is gonna shift it to the right. Why? Because we said right shift is because decrease in affinity, and also it's acidic. And the same goes for this one and this one. It's just the combined effect. Now, the next thing we're gonna look at is temperature. How does temperature affect the curve? When the temperature increases, the O2 affinity will decrease. This is important for us because this is how the body gives us oxygen in times of need when we're working out, when we really need it. Because let's say you're exercising. When you're exercising, your muscles warm up, right? So you're gonna have an increase in temperature in your muscles. So whatever blood is flowing by over there, your temperature, the increase in temperature is gonna decrease the affinity. And decrease in affinity basically means what? The red blood cell is gonna let go of the oxygen, which is good because we need to take up this oxygen and use it because we're working right now, so we need it. Thus, we would have more oxygen available for use. So 41 degrees increase in temperature, it's gonna shift to the right, and that's gonna give us more O2 for the tissues to work. Then we have another thing that affects the curve, which is BPG. Full form is biphosphoglycerate. And it's got another name. It's also known as 2,3-DPG. They're the same thing. It's just 2,3-bisphosphoglycerate. And they also alter the affinity for oxygen. How? If BPG concentration increases, then you're gonna have a decrease in affinity. So you see how most of the things, when they increase, they decrease the affinity, right? H plus increases, it decreases affinity. CO2 increases and decreases the affinity. Temperature increase, decreases the affinity. And now even BPG, when it increases, it decreases the affinity. Okay, what is BPG? BPG is something that our body is going to produce in the case of chronic hypoxia. So when we have hypoxia, which means less oxygen, BPG is going to go, it's going to go to the hemoglobin, it's going to bind here, and it's going to bind to the point where you see how it's so far we've been seeing just one oxygen being left, but here we have like 4 oxygen molecules that are just being released, which is good because it's hypoxia. We want the red blood cells to let go of this oxygen. Because remember how we said, physically dissolved oxygen is the one that's important. It's the one that needs to go through the membranes and go to the tissues for it to be used, right? So we want this oxygen to leave the red blood cell and go somewhere. This could happen in the case of high altitude when you go to someplace high up, or if you have a chronic lung disease and you have less oxygen. This is what it does, right? And it says it assists in the unloading of of O2 to the peripheral tissues. Now, we've been saying it binds to this, it binds to that. What is that? What is it binding to? So we have, this is hemoglobin. You don't need to really know the structure, but it's just to make you understand a little bit more. When we say it's binding to the beta chain, it's just these. If it's binding to the heme, it's this yellow part. And yeah, this is just the chemical structure of heme. You don't need to know any of this. Okay, now we have something called carboxyhemoglobin. What is carboxyhemoglobin? It is when hemoglobin will bind to carbon monoxide. We call it carboxyhemoglobin. It's as simple as that. So we have 3 scenarios here. The first one is hemoglobin with 100% saturation, and it's loaded with oxygen. It's normal. There's nothing wrong. This is how it is normally for us. Now, if we have anemia, 50% anemia, what is 50% anemia? It means that we have half the amount of blood, let's say, okay? It's still 100% saturated. It's still 100% saturated. It's just that it's less. The concentration is going to be less. We're going to have less amount of oxygen in the blood. Lastly, we have what we're going to talk about. It's carboxyhemoglobin. When carbon monoxide binds to the oxygen, to the hemoglobin, okay? So carbon monoxide, when it binds to the hemoglobin, it's going to increase the affinity for oxygen. We're going to see it a little bit more, but it's just 3 scenarios, and we're going to look at the graph for this later on. Carbon monoxide poisoning. So this is the graph, the 3 graphs for everything that we've seen before. So normal blood is the normal graph that we've seen. And then we have the blue curve, which represents anemia, 50% anemia. And the number of red blood cells are halved, right? So that's what we were talking about before. If the hemoglobin is halved, the concentration is going to be affected and it's going to decrease. See? Carbon monoxide, it's kind of weird, but you have to bear with me. It's not hard. It's just you need to just get it in your head. Carbon monoxide, it can reversibly bind to the hemoglobin. Okay. However, the thing about carbon monoxide is that it has a very high affinity to the hemoglobin, and then as soon as it binds to the hemoglobin, it will increase affinity for oxygen. So it's not going to take up all the binding spots. It's gonna like go and bind to some, And then the rest of it, it's gonna call the oxygen to stay with it. Okay? So that's what's happening in this part, basically. You see how this graph is going this way, this graph is also going this way, but this green one, it's a little bit more upward, right? Basically, the concentration of O2, it increases. I'm so sorry. It increases really quick because as soon as the carbon monoxide comes and binds, it's gonna bring the oxygen also to come and bind with it. Okay? So, in the case of carbon monoxide poisoning, just give me a second. Okay. In the case of carbon monoxide poisoning, the thing is, we don't need a lot of carbon monoxide to have an adverse effect, okay? Even if you inhale, let's say about 0.1% of carbon monoxide, you're gonna have 50% of your hemoglobin just bound to the carbon monoxide, okay? So when we look at the pressure of the gases, the pressure of oxygen is still gonna be normal. That's what it says. During carbon monoxide poisoning, the PO2 and the hemoglobin concentration may be normal, okay? But the O2 concentration is less. Why? Because half of them are just bound with the carbon monoxide himself. Severe carbon monoxide poisoning may require blood transfusion or treatment with hyperbaric oxygen. What is hyperbaric oxygen? It's just we give the patient higher pressure of oxygen because we said carbon monoxide, it reversibly binds. It's like a competitive inhibitor. So when you increase the pressure of oxygen, you give out more oxygen, it's gonna go and take it out of the place. Okay? So far, any questions? Do you want me to repeat anything, explain anything?
Yes, hello.
Yeah?
Earlier you were explaining why what's it called in the graph there's like 0.75% hemoglobin at like 40 PO2. I
Mm-hmm.
didn't really understand that.
Oh, okay. That is when I was explaining the graph.
Oh yeah.
Okay. Okay. Basically, what I was saying is if we look at the partial pressure of oxygen and we go to 40 millimeters of mercury, where do we find this pressure? We find it in our venous blood. Okay. So the blood that goes after it's been used by our organs, after all of that, when it goes back to the lungs from our body after being used, it still has 75% saturation. What is 75% saturation? It means that 75% of the hemoglobin is still bound to oxygen, which means we have a lot of oxygen still in our blood, right? So why do we have that? We have two reasons for that. One of them is very, very simple, very common. Just very general. It's just that our body, it's giving 75%, it's leaving 75% of oxygen at rest. As in, you're just normally working, you're sitting, you're just doing all of that, and you still have 75% of oxygen in your body. Why? In the case of, let's say, an emergency, I want to get up and run, I need extra oxygen, like instantly. I need my muscles to use this oxygen instantly, right? So we have this extra oxygen in our blood, which is a good thing. So we can use it in the case of exercising, in the case of an emergency, anything that would need us to use more oxygen than normal, we have it in our blood. Another reason that we have so much oxygen in our blood is because different parts of our body are going to use different amounts of oxygen. So if we look at anything, the example I use is the heart. It's basic. The heart is always pumping, right? So it's gonna consume a lot of oxygen. So the saturation of the blood that's coming from the heart is gonna be really low. It's not gonna have that much oxygen. But what's the point is, it's gonna come in the same place. All the blood is coming in the right ventricle. So 40% saturated blood is coming to the right ventricle, along with something else, some other place, the kidney, that's not using a lot of oxygen. It still has like above 90% oxygen in the blood. They're all coming and they're mixing in one place. Okay, so essentially, you're gonna have around 75% of oxygen. So when we say deoxygenated blood, you can't think there is no oxygen. It's just less oxygen. We still have at least 75% oxygen in the blood. Did you get it?
Yes, thank you. Clear.
Welcome. Anyone else? Any other questions? Should I just move on? Because if you don't have questions, I have questions. Okay.
There's a question in the chat. Someone is asking to recap the carboxyhemoglobin.
Sure. Okay, so how do I erase this again? 1 second. All right. Carboxyhemoglobin is basically hemoglobin bound to carbon monoxide. We went past that. So this is just three. I don't know why anemia was also here, honestly, but it was put here with the carbon monoxide. So anemia, it's simple. It's just this. So let's leave that aside. Carbon monoxide graph, most of the graphs, they start like this. They curve like this, and then they go up. Carbon monoxide, it starts this way. What does this mean? If you look at the value here, let's say, at this low pressure, maybe 15, maybe 20, we have so much concentration of oxygen already, right? But we said that carbon monoxide is binding to the hemoglobin. So how do we have this increase in the O2 concentration? Basically, when the carbon monoxide, it binds, I'll just remember this picture. Carbon monoxide are these red ones, okay? When they come and they bind, they're not gonna bind and take up all the sites. They're just gonna take those two sites, and then as soon as they bind, they will let the hemoglobin take off. They're gonna increase the affinity even more than normal. They're gonna basically the amount of affinity that the hemoglobin might have normally had, it's gonna be even higher. Okay, it's like a fake friend. It's like come, we love you, and then they're not gonna let you go. It's like that. So carbon monoxide, it'll bind, and then as soon as it binds, a lot of oxygen also binds. Okay. However, this graph is about the O2 concentration. Okay. So it goes like this, the O2 concentration, and then it stops. It becomes steep. Why? Because 50% is carbon monoxide bounded. So only 50% is allowed to bind with the only 50% hemoglobin is allowed to bind with the oxygen. So it kind of, you see how the values, they kind of merge with the half anemia, 50% anemia, and the carbon monoxide, 50% carbon monoxide. The only difference between these two graphs is this part and this part, the one that takes oxygen all of a sudden. Is that okay?
Yes, it's good.
Thank you. Okay, so I have a few questions. They don't have MCQ. I just need simple answers. I just want to know if you understood it. Okay. So what is the effect of pH on the oxygen binding curve? I just want to know what happens when it increases, what happens when it decreases. Anybody, just say it. It's okay if you're wrong. Increase in pH with left, shift to the left. Yeah, that's right. What about decrease? Perfect. Okay. How does CO2 indirectly affect the affinity? What does the CO2 do that will go and decrease the affinity? Yep, that's right. CO2 decreases the pH, so it increases the hydrogen ions, and that in turn decreases the affinity. Perfect. What would lead to an increase in the concentration of BPG? Yeah, high altitude hypoxia, that's right. Or if you have a chronic lung disease where you don't have enough oxygen, right? Lastly, how does carbon monoxide, what does it do? Increase or decrease affinity of hemoglobin towards oxygen? Yeah. Okay, these were really simple, but I want you guys to like solve a question. So, you know, this is what you would get in the exam. Does anyone want to read, or I'll just read the question? Okay, I'll read it. A 26-year-old woman presents to the urgent care facility with rapid and shallow breathing for an hour. She describes having an anxiety attack and cannot catch her breath, and complains of dizziness and tingling in her hands and feet. Her heart rate and blood pressure are slightly elevated, and blood temperature is normal. Which of the following changes is expected in this patient? A left shift of O2 dissociation curve, right shift of O2 dissociation curve, increase in plasma bicarbonate, decrease in arterial PO2, or an increase in arterial PCO2. Okay, why do you think it's E? We said she's having rapid and shallow breathing. Yeah, you're right. It's A. Okay, do you want to tell us why it's A? Yeah, good job. Perfect. So we're saying that this woman is coming with rapid and shallow breathing, right? So she's having hyperventilation. Another thing, I'm pretty sure it's not said, the basically dizziness and tingling, they're kind of signs of alkalosis. And alkalosis is what? Increase in pH, right? So both of these things, hyperventilation and increase in pH, they're gonna give you a left shift of the dissociation curve. And these are just to remind you what are the things that give you a left shift. And if you guys want to know why it's not the other options, I wrote it in the notes, but if you want me to tell you, I can tell you right now. I'll just say it. It's not a right shift, obviously, because there's nothing that is going to reduce the affinity, right? She's hyperventilating. Hyperventilating is when you're not even letting the carbon dioxide, you're not letting anything happen, you're just breathing in and out, in and out. So you're not increasing PCO2. So E is out, right? And decrease in arterial PO2, no, because you're hyperventilating, you're breathing in a lot. So it's not a decrease either. Nothing to do with bicarbonate. And since it's rapid and shallow breathing and alkalosis, it's a left shift, so it's not a right shift either. It's really easy. Okay, moving on. Do you guys want a break? Or should I just continue? Okay, because we're halfway there, so if you want to pray, I can wait for 10 minutes. We can be back. Okay, 5 minutes. You can go pray and come back. Is that okay? Because it's 5:45. Okay. So now that we're done with the curve of oxygen, one last thing about oxygen, and then we're gonna move on to carbon dioxide. Okay. So oxygenation and oxidation, these both happen in the hemoglobin, and it's really, really easy. Basically, oxygenation is when the O2 is gonna go to the hemoglobin, bind, go to any tissue, leave it out there, and then come back. Okay, this is oxygenation. Oxygenation is basically the process which consists of loading the oxygen and unloading it. So it's just binding and unbinding. There's no changes that occur in the heme itself. Oxidation, however, is different. So heme is normally present as Fe2+. Now, for any reason, if you have an oxidizing substance that will come and oxidize the hemoglobin, we're going to form something which is called as a methemoglobin. A methemoglobin is basically when the iron has Fe3+ now instead of just 2+. So you see how here, as soon as it becomes Fe3+, you're not going to be able to bind. It's blocked, basically. You can't have any oxygen coming and binding here. And therefore, if you have methemoglobin, you can't transport oxygen at all. And methemoglobin is what? It's just oxidized hemoglobin. It's called methemoglobin. And normally, we have some amount of hemoglobin in our blood that can get oxidized because we have a lot of substances in our body, right? But it is brought back to normal. How? It's just reduced oxidation reduction. So methemoglobin is reduced back to deoxyhemoglobin or normal hemoglobin. And we do this by an enzyme. And this enzyme has a very obvious name, methemoglobin reductase. So it's going to bring back the Fe to the Fe2+ form, and it's going to be normal hemoglobin again. Okay? So how do you remember this is oxygenation and this is oxidation? The way I like to remember is oxygenation has the entire word oxygen in it. It likes oxygen more, okay? So it helps the oxygen. It takes it here, takes it to, let's say there's some tissue here, and then it'll go, and it comes back. Okay, oxidation is the bad one. We're gonna have something come, and then it's not gonna let oxygen come with it. Okay? Now let's move on to carbon dioxide. So we said oxygen has 2 forms in the blood, right? Our carbon dioxide has 3 forms in the blood. One of them is the simple physically dissolved form, and there's 2 chemically dissolved forms. So physically dissolved, I know the name sounds complicated, but it just simply means the free molecules of the gas. So this one is a physically dissolved. It's just simply CO2 in our blood. Chemically, we have bicarbonate and we have carbamate. Another name for carbamate is also carb amino CO2. Okay? What is bicarbonate? Whenever we have carbon dioxide in the body, it will react with water and it's gonna form bicarbonate. And carbamate is basically CO2 will directly go to any protein. Okay? So this is just representing any protein. And what is the protein in our blood? Hemoglobin. Hemoglobin is a protein, okay? So it'll just go and it'll bind, and we call that carbamate or carb amino CO2. Now, the one that we have a lot in our body is bicarbonate. So 70% of carbon dioxide in our body is in the form of bicarbonate. And then we have 20% in the form of carbamate and only 10% in the physically dissolved form, okay? So we have all these reactions happening, right? CO2 plus H2O, CO2 and proteins. Let's go more into that. So reactions of CO2. So we have this, it's any tissue or any lung producing CO2, okay? So CO2 is produced due to metabolism. Any activity is gonna produce CO2 in our body, and it's gonna go from the tissue into the plasma, and then from the plasma into the erythrocytes. In the erythrocytes, some of the CO2 will bind with hemoglobin. Hemoglobin is a protein. What is it going to form? It's going to form carbamate, okay? In the plasma and also in the erythrocytes, so this is happening in both the places, we're going to have CO2 reacting with water to form bicarbonate and H+ ions, okay? So that would be here and here. So these H+ ions that are produced because of bicarbonate, you see it's bicarbonate plus H+ and bicarbonate plus H+. They need to be bound to a protein. Why? Because we don't want increase in H+, right? What does increase in H+ do? It decreases your pH. It causes acidosis. So we're going to have to buffer this H+. In the plasma, we have buffering proteins. It's represented by a PR over here. And in our erythrocytes, again, what is the protein? Hemoglobin, okay? So H+ is gonna go bind to hemoglobin. So in the plasma with buffer proteins and in the erythrocytes with hemoglobin as the protein, okay? So there is a difference in the bicarbonate production in the erythrocytes and the bicarbonate production in the plasma. Okay, why is that? First of all, in the erythrocytes, we have a lot of hemoglobin. Okay, so when we have bicarbonate production, when we have this reaction taking place, we have this H plus. H plus is going to keep increasing, right? And if we have a lot of protein that is ready, it's ready to take the H plus and buffer it, this reaction is going to occur more, right? So we're going to do what essentially? We're going to keep increasing the bicarbonate. So compared to this bicarbonate and this bicarbonate, this one in the erythrocyte is gonna increase more. Why? Because we have more protein available to take the H plus away from it and buffer it. Okay? So buffering capacity in the erythrocyte is much higher than the plasma. Another thing we have in the RBCs is this guy over here, carboanhydrase. What is carboanhydrase? It's an enzyme. It's an enzyme that is going to facilitate the reaction of carbon dioxide reacting with water to give us bicarbonate and H plus ions. So it means there's two reasons for more bicarbonate production in the erythrocytes. We have more buffering capacity and we have an enzyme, carboanhydrase, that supports this reaction. So we have faster and more bicarbonate production. What is the significance of knowing this? Basically, when you have a lot of bicarbonate concentration in the erythrocyte, essentially it's going to want to move out, okay? So when it goes out through this one is a transporter, and this transporter basically exchanges the bicarbonate with the chloride ions. So as this reaction keeps taking place, it's not instantly thrown out into the plasma. It kind of happens slowly over time, okay? So you're increasing bicarbonate, and then eventually it's going to start moving out and exchange itself with the chloride ions. Now, as it keeps moving out, at some point, you're going to have a really high concentration outside. So what it's going to do is this green arrow over here, it's going to start coming back inside. It just does that. So now it's going to exchange with the chloride ions again, but this time chloride ions are going to move out of the cell and bicarbonates are going to move back into the cell. And this entire phenomena is called hamburger shift. I know it's a weird name, but it's just called the hamburger shift. Okay? Any questions in this? I can go over this again. Anything? Do you want me to repeat? Do you want me to do the entire thing again? Okay. So we said there's three forms of carbon dioxide, right? We're gonna look at the chemical ones over here. We have bicarbonate and we have the carbamate, which is bound to the proteins. So when we have CO2 that comes out of the tissue or the lungs, it's gonna, oh, why is it black? It's gonna go and it's gonna go to the plasma, and then from the plasma, it's gonna go to the erythrocyte. So first here and then here. Now it's gonna do two things. In the plasma, we said carbamate binds to a protein. What is that protein? It's hemoglobin. So we're done. This is one of our chemically bound carbon dioxide. It's just carbamate. Another thing is we said that CO2 will react with water to give us bicarbonates. Now, the thing about bicarbonates is that there's a difference in the bicarbonate production in erythrocytes and in the plasma. In the erythrocytes, we have way more bicarbonate. Why? Because of two reasons. In this reaction, along with bicarbonate, what are we producing? We're producing H plus, right? H plus is it's gonna cause the entire thing to become acidic, and we don't want that in our body. So in the plasma, when we release H plus, we have these buffering proteins over here, and they're gonna just, what's the word? Yeah, buffer, because they're buffering proteins. And the protein that we have in the erythrocyte is, again, we saw hemoglobin here, it's hemoglobin again. So the H plus and hemoglobin, they're gonna buffer. But in our erythrocytes, this is a lot. We have a lot of hemoglobin, okay? So as soon as this reaction takes place, this one, it goes and it gets buffered. So it keeps happening faster and faster because we're getting rid of this H plus faster and faster, okay? Another thing in the erythrocytes is we have an enzyme, the carboanhydrase. Carboanhydrase, it facilitates this reaction. It makes this reaction happen even more. It's like encouraging it basically, right? So what happens is we have way more of this thing happening. So every time CO2 comes, this enzyme is there and it's like, yo, go react with the water, and it makes it happen. So we have faster bicarbonate production and more. Like there's more in number as well. So what's the significance of this high amount of bicarbonate? It's basically this high concentration of bicarbonate at some point is gonna want to leave because there's a lot. So it's like, I want to go. It's gonna go out through this transporter, which is an exchanger. So one bicarbonate goes out, one chloride ion comes back inside. So this keeps happening, and this is not very fast, right? So eventually bicarbonate keeps moving out one by one, and then eventually what you're going to end up doing is you're going to end up increasing the bicarbonate in the plasma. And this increase is not going to happen because of this reaction. It's going to happen because this one keeps moving out, okay? Now, at some point, it's going to be a lot in the plasma as well. The concentration is high in the plasma. It's going to go back in. That's the green arrow, this one. So it goes back inside. And like we said, it exchanged itself with a chloride ion, so it's going to do the same again. Chloride is going to move out, bicarb is going to move in. And this shifting of the bicarbonate going from inside to outside and then outside to inside is called as a hamburger shift, literally hamburger. That's it. Was that okay? All right. So we saw there are factors that, we have the oxygen curve, right? Similarly, we have the CO2 dissociation curve. So this is a CO2 dissociation curve, and basically we have similarly the physically dissolved here, and it's very little, it's just this much. We said it's how much? 10%. And then we have this one. This one is for the chemically bound CO2. So the physically dissolved has a linear graph, which is this purple line. And similarly, as the PCO2 increases, the concentration is also going to increase. Now, if we look at the total concentration of CO2, okay, we will see that a slight change in PCO2 can increase CO2 concentrations dramatically. So remember our oxygen curve, it would go like this, and then it would go, and then it would become flat, right? That's it, nothing. But here, we have this one going like this, and it's very steep, right? What does it mean by steep? If you take even the tiniest of pressure differences from here to here, and you go all the way up, you're going to see that it's going to have huge concentration differences from here, okay? So in our oxygen, it was binding to the hemoglobin, and you had saturations and stuff, and it would become flat. But here, the chemically bound, it can keep increasing, okay? So remember when we said when we add more oxygen to the oxygen binding curve, it's going to start adding into the physically dissolved graph, and the rest of it is just going to parallelly follow. But here, this, you see how this graph, it's going to keep going this way, okay? The difference between these two is the amount of the chemical form, and we know that. So we saw the factors that affect O2 binding curve. Now we're going to look at the factors that affect the CO2 binding curve. It's not a lot, and it's honestly kind of similar. So first we have the H plus ions. Now, H plus, where do we get the H plus from? We get it from carbon dioxide and the water reaction, right? So when there is an increase in the H plus ions, they will go and they will react with the bicarbonate in the blood. So here, instead of this reaction happening first, this one's happening first. So we're adding H plus. So H plus is gonna make this reaction go in this direction, and you're gonna have more H2O and more CO2. What happens to this CO2? This CO2 is gonna just be released, basically, okay? So we exhale this carbon dioxide and we lose it. Essentially, what's happening? What is this bicarbonate? This bicarbonate is our chemical form of CO2, right? So if we put H plus, it dissociates and just loses this carbon dioxide. We're losing our carbon dioxide. So you see, this blue one, this one's normal, and this red one is what happens with the increase in H plus, because the concentration is going to decrease, okay, because we keep losing the CO2. So this is going to shift the curve to the right since concentration of CO2 decreases. This is not going to go into physically dissolved, basically. Another thing, it was just written as a sentence. So in your slides, it was just a little bit. It's just that increase in temperature also shifts the curve to the right. It's kind of similar like the oxygen. Increase in H plus ions is going to shift the curve to the right. Increase in temperature is also going to shift the curve to the right. Okay. There's something we have here which is called the Haldane effect, and it's kind of similar to the Bohr effect. Okay. So when the oxygen binds to hemoglobin, the affinity of hemoglobin to bind to CO2 decreases. So what is CO2 bind with hemoglobin? It's the carbamate, the carb amino CO2 that we had, right? So whenever we have this hemoglobin and the O2 comes and binds, it's gonna let go. The CO2 is just gonna be thrown out because the affinity is gonna be decreased towards CO2. This is the Haldane effect. That's literally the Haldane effect, okay? What about the Bohr effect that's here? Because these two go hand in hand, okay? Bohr effect is where an H plus ion, which is produced because of CO2, we've seen that in the oxygen, and we've seen the reaction before. So H plus is gonna, H plus that is produced because of the carbon dioxide will bind to the hemoglobin. And we said what? It causes a conformational change, right? The conformational change is gonna do what? It's gonna decrease O2 affinity. So you let go of the O2. It's not bound to the hemoglobin anymore. Where is this significant to us? When the blood comes to the lungs, the hydrogen is going to leave its binding site from here, okay? It's bound here usually, but it's going to leave, okay? It's not there in your lecture, but I'll tell you why it leaves. Basically, when the blood comes into the lungs, we're trying to get rid of the carbon dioxide, right? And we said only 10% is a physically dissolved. So how are you going to get rid of the the chemical form? This H plus will leave the oxygen. It will react with bicarbonate. That's how it'll go do that work. And it's going to give us the CO2, which we need to exhale, okay? So when the H plus leaves its site, what's it going to do? Decrease in H plus concentration, so higher affinity for oxygen. So when you're in the lungs, this is going to increase affinity of the hemoglobin, which is good. You want to load the hemoglobin with the oxygen in the lungs, which is this Bohr effect. It's O2 loading in the lungs. Now that the oxygen is bound to the hemoglobin, the affinity of hemoglobin for CO2 decreases. So the CO2 is then released to go into the alveolar gas. Sorry. So this one was this H plus leaving was for the bicarbonate. What about the carbamate? How are we going to get rid of the carbamate form of CO2? Now, when the oxygen binds, it comes to its side. We inhale, the oxygen came and it bound to this hemoglobin. Now we said CO2 affinity of hemoglobin to CO2 will decrease. So it lets go of this. And then we exhale. Okay, so both Haldane effect and Bohr effect, they help each other. Okay, first Bohr effect happens, and then the Haldane effect happens. So Bohr effect, it's loading in the lungs, O2 loading in the lungs, and Haldane effect helps in CO2 unloading in the lungs, which is our respiration. And then we have in the systemic capillaries, it's the opposite, right? Because we want to take away the carbon dioxide and we want to give it the oxygen. So O2 unloading in systemic capillaries and CO2 loading in the systemic capillaries. Okay, do you want me to repeat? Do you want me to go over this again? Or if you want me to go over the curve? Nothing? Not at all? Is everything clear? Okay, that's great. Okay, so we're just gonna summarize real quick. Okay, this is for the O2 curve. What about hydrogen? When hydrogen concentration increases, affinity is gonna decrease. And this is the Bohr effect. It's part of the Bohr effect. Now, CO2, when CO2 concentration increases, affinity is going to decrease. What is affinity decreasing? It's the curve shifting to the right. Okay. BPG increases, affinity decreases, curve shifts to the right. And this happens when in chronic hypoxia. Temperature increases, affinity is going to decrease. This is significant to us in exercise when our muscles need work. And there's one more thing. It's very, very simple. Type of hemoglobin. So there can be various types of hemoglobin as well. For example, fetal hemoglobin has higher affinity than maternal hemoglobin. And I mean, it's obvious, right? It's a baby. It's going to have higher affinity for the oxygen. Okay. And it's just in the amino acid sequencing of the hemoglobin. And then we have carbon monoxide. It increases the affinity of the hemoglobin, but since the carbon monoxide itself binds to the hemoglobin, it decreases the oxygen concentration. Then we have our factors affecting CO2 curve, temperature. Temperature increases, affinity decreases, curve shifts to the right. And Haldane effect is what? When oxygen binds to hemoglobin, carbon dioxide affinity decreases, right? So that's that. Concentration of oxygen increases, affinity will decrease. And H plus, if it increases, affinity will decrease. So yeah, that's it. We're done with the lecture. And if you guys are good with everything, we're gonna do some questions. Okay? So this is the first question. A 41-year-old woman is receiving mechanical ventilation following a drug overdose. On the fifth day of her admission, she develops a fever and is found to have a bloodstream infection. The arterial blood gas that morning had an arterial PO2 of 72 millimeters. Normal is 75 to 100. That was unchanged compared to the blood gas results from the preceding day. So we see that her PO2 is low. Which of the following physiologic changes would you expect? A, there's a decrease in carbon dioxide production or a decrease in shunt fraction, increase in arterial oxygen concentration, increase in arterial oxygen saturation, or increase in P50 for hemoglobin. Mm-hmm. Yeah, that's right. So it's E. And this is why. We see that she develops a fever, so she's got an increase in temperature, and her arterial PO2 was also less. Does anyone want me to explain why the answer is E? Or should we go to the next question? Okay. So you have this curve. In the hemoglobin oxygen dissociation curve, oxygen dissociation curve is the same as the oxygen binding curve, so don't get confused. The shift from curve A to B is caused by increased pH, decreased BPG, strenuous exercise, fetal hemoglobin, or carbon monoxide poisoning. Okay. Can anyone tell me why it's C? Yep, it will be increase in temperature. So the curve is going to shift to the right. Increased pH would go to the left. Decreased BPG would go to the left. Fetal hemoglobin would go to the left. And carbon monoxide, we know it has the little curve, the extra curve in the bottom, right? Okay, next question. A man fell asleep in his running car. He was unconscious when he was brought into the emergency department, and he's got carbon monoxide poisoning. You would expect his alveolar partial pressure would be what, and his arterial O2 would be what? Okay, anyone else has another answer? Okay, so you guys are right. It is normal and decreased. Also, the thing that I talked about, 0.1% carbon monoxide, that is enough for carbon monoxide poisoning, because if there's really, really high, it could be lethal, okay? So yeah, that's why very little could cause carbon monoxide poisoning, and that's why alveolar partial pressure is usually normal. Okay, we have one last question, and it's about graphs. Which points would represent arterial blood in a severely anemic person? So one graph is the concentration, right? This is the concentration, and this is the saturation. Anybody else? Okay, well, the answer is D. Does anyone want me to explain why it's D? Are we all sure why it's D? All right. So we have one graph that's talking about the concentration, right? This is telling us how much oxygen, and this is telling us what the ability to bind. So remember my diagram, which was like this, and well, it's not mine, it's Dr. Shams, but it was like this, the four little circles, and one was like this with two circles, and then they had hemoglobin bound to them, right? So you get the point. So all this hemoglobin bound to them. This is normal. This is a normal person, right? And their concentration of oxygen, these oxygen molecules, it's gonna be high, right? So it's gonna be somewhere here. But in this one, we have less hemoglobin molecules. Okay? We don't have all of these, we don't have a lot of hemoglobin molecules. We only have two of them here. But if you look at the content, how much is there? How much deciliters are there? You're gonna be here because you're not gonna have your normal concentration of oxygen, right? What is anemia? You have less blood, and less blood means less O2 concentration. However, we said bottom graph is TD. Why? Because this is saturation, which means that this molecule, even though it's less, it's still binding all four oxygens. Okay? So even if there is less hemoglobin, the ability, the amount of oxygen it can take. That's saturation. So it's still normal blood cells. It's just lesser blood cells. So this one is going to be affected. Concentration is going to be affected. However, this is going to remain normal. The saturation remains normal and anemia, okay? Okay, great. If anyone wants me to go over anything in the entire PPT, you can tell me now. And if not, then you gotta fill this form. It's just one question, guys. So please fill this before you leave. Thank you. If you guys have any questions, feel free to contact me, okay? Thank you.